Well, thanks very much, Lee, for that really nice introduction. And thanks to the Tanner family for making this entire event possible. And thank you so much to all of you in the museum and in the integrated biology for your hospitality. I'm having just a great visit, and I look forward to talking to more of you tomorrow. Let's see. So what I'd like to talk with you about tonight is some of the research that Doug and I have been pursuing on speciation in the plant genus Tragopogon. Now, if we think about how Darwin thought about speciation, and in fact about how probably most of us think about speciation, we tend to think about species forming through a slow, gradual branching type of process. But instead, the examples that I want to share with you this evening represent a very different mode of speciation, and that involves rapid generation of new species and reticulation. So instead of species arising through a branching divergent process, speciation actually occurs by bringing the genomes of two different existing species together. And this is through a process known as polyploidy. And before we can actually understand how all of this process works, we need to have a few definitions. So, for example, first of all, a diploid organism or cell contains 2 copies of each chromosome. In contrast, a polyploid has 3 or more copies of each chromosome. And there are 2 main classes of polyploids, autopolyploids, and we won't really be focusing on those this evening, but these are formed from within a single species. This is a very generalized definition. And in contrast, then an allopolyploid, which combines the genomes of more than one species. And this allopolyploidy is diagrammed here, and this occurs when we have a diploid species, here diploid A with, in this example, 8 chromosomes, diploid species B with 12 chromosomes. These 2 species hybridize, and the genomes come together to form a single genome that has all of those 8 chromosomes from diploid A and the 12 chromosomes from diploid B. So this would be referred to then as an allopolyploid, something that has doubled the chromosome number of the parental species, and that the species arises by bringing these 2 different genomes together. Oops. Now, polyploidy is typically thought of as being sort of a plant thing, but what I'm going to try to convince you of tonight is that even though it's very important in plant evolution, polyploidy is also prominent in other major groups of organisms as well. So in plants, we've known that it's very significant in terms of the overall evolutionary scheme of things for quite a long time. And traditional estimates suggest that maybe 50% or more of all flowering plants or angiosperms are of polyploid origin. And for those of you who aren't familiar with plant systematics, the flowering plants represent about 250 to 300,000 species, and they're, of course, the most speciose, the biggest group of plants. Also, it's thought that about 95% of all ferns may be polyploid. There are 17 of the 18 world's worst weeds in a categorization that was made, this was about 20 years ago, were considered polyploid. And it may be, in fact, that all 18 of these are polyploid, but that 18th one just hadn't had its chromosomes counted yet. In addition, all of our major crop plants are polyploid, with just a listing shown here of some of these. So there's tremendous economic importance associated with polyploidy, both from a positive perspective in terms of crop improvement, and also negatively because these polyploid species seem to have a propensity to become serious weeds. So understanding something about polyploidy and its genetics and its evolutionary consequences is important for a number of reasons. Now, polyploidy may be as simple as the example that I gave you before, where you just take 2 species and they hybridize and you get a doubled chromosome number going from 8 and 12 up to 20 pairs of chromosomes. Or polyploidy in some lineages may be much more extensive. So this is just a sample of the types of chromosome numbers that you may find within a single genus of plants. So in the genus Sedum, this is a rock garden plant. There are some species with 16 chromosomes and then all the way up to 640. You can see some of these other examples. Within the single species Claytonia virginica, the chromosome numbers range from 12 to 192. And then our favorite one is the species of Ophioglossum. This is a fern species that has 1,260 chromosomes. And you wonder, first of all, why does a fern need all of those chromosomes? And secondly, how on earth do they ever get through their cell divisions with all of those chromosomes to deal with? Now, there are a number of things that one generally learns about polyploidy when you go through school. And so some of these traditional views of polyploidy are listed here. First of all, polyploidization was considered rare. So even though there's a huge proportion of plant species that are polyploid, the idea of getting a polyploid species in the first place was considered quite rare. And again, this was something that was limited to plants, and so big chunks of biologists weren't really interested. Autopolyploids, I said I wouldn't really mention them, but they are considered to be rare relative to allopolyploids, and I would say that that is probably still true. Another thing that we generally thought about polyploids until Charlie Worth's really great paper on fern polyploidy back in, I think, 1985, was that each polyploid species had a single origin. And this will, the opposite of this, the converse, will become apparent in a few minutes. But the idea here is that if a species just formed this one time through this hybridization event, all of the individuals that descended from that speciation event would be genetically uniform. And in addition to that, because there are multiple chromosomes, multiple genomes within the single composite genome of the polyploid, there's a buffering effect. So if any new beneficial mutation arose, it would be very difficult to get that mutation incorporated into the genome and have that expressed because there were all of these extra chromosome sets. So for these reasons, the genetic uniformity and this buffering effect, polyploids were actually considered to be evolutionary dead ends. Again, despite the fact that so many plant species are known to be polyploid. And this was the view of such prominent plant evolutionary biologists as Herb Wagner and Ledyard Stebbins. Now, some really amazing work started coming out of the genome sequencing of Arabidopsis. And this is what started to bring polyploidy into a realm of consideration outside of just plant systematics or plant evolutionary biology, or in some cases, the crop biology groups. So, whoops, wrong button. So, of course, Arabidopsis was selected for sequencing because it had a small genome. But the amazing thing was, as this genome was beginning to be assembled, it was determined that there were, in fact, 2 or maybe 3 rounds of genome duplication, in other words, polyploidy, somewhere in the ancestry of Arabidopsis. And that's shown in this phylogenetic tree here that shows that there was a genome duplication event at some point here several million years ago that was shared by the genus Brassica, which contains a lot of vegetables like broccoli, and it has rapeseed and so forth in it, and Arabidopsis. So there was one genome duplication event there. There was another one that was dated back to this point, which is somewhere around maybe 120 to 150 million years ago. And this at a point in plant evolutionary history that contains, well, that is the common ancestor for about 75% of all of the flowering plants. And there may have been yet another genome duplication event that occurred prior to the evolution or the origin of the flowering plants. So based on a number of different sorts of analyses, and I've just listed a couple of them here, Arabidopsis, this model plant for the study of genome structure and genetics, actually turns out to be a polyploid. Oops. There we go. Now, just a few years ago, a really interesting paper was published by Blanc and Wolf in which they used a genomics approach to go into all of this vast genomic data that are being generated. And they used this concept, and I won't go into the details of it, but the concept of this characteristic peak in the frequency distribution of duplicate gene pairs. In other words, they took genomic data for species such as maize in this case and cotton in this case, and they looked for evidence of a peak in this frequency distribution. And this peak is a signature for a genome duplication. And it was really, so they tested this approach on things that were known tetraploids. So we knew that maize was a tetraploid, and we knew that these cotton species were tetraploids. And they showed these peaks. So the peaks vary in size, but that peak still occurs in that location. So this gave them confidence then to apply this technique to other groups of plants for which large genomic resources were available. So they did this, and they found that everywhere you see one of these ovals, and don't worry about the different coloring of them, but everywhere you see those ovals, there's actually evidence of a genome duplication, or in other words, a polyploidization event. And if you'll notice here, whoops, did it again. Most of these actually represent economically important plants. Number of actually all of them, crop species, and then our Arabidopsis model down here. So what this indicates is that all of these plant species have undergone genome duplication, and in fact there's even evidence or inferences that there were genome duplications in the ancestors of some of these different lineages. Well, a few years ago then, shortly after we saw the results of that study, we thought, wow, this would be a really interesting thing to do, not just with crops and economically important plants, but with some plant species that are evolutionarily important. And so we picked a number of species that occur at key locations in the family tree of the flowering plants, and we applied the same technique using some genomic resources that we developed. And so just for example, this is a monocot. Its name is Acorus. This is a poppy. This is avocado. And then this is a water lily. And all of these occur at key locations in the phylogenetic or evolutionary tree for flowering plants. And all of these, boy, sounds like they're having fun. All of these show some sort of peak here early on in this distribution. And what this suggests then is that there were, in fact, genome duplication events clear back in the ancestries of these lineages, most of which, this one, and this one at least, are some of the most ancient lineages of flowering plants. So what this suggests to us is that maybe we need to revise our estimate of polyploidy in the flowering plants. Maybe we shouldn't argue over whether it's 48% or 55% or whatever the scientists of a few decades ago were arguing about. But in fact, when we use these genomic tools and we look at the possibility of polyploidy, we see, in fact, that nearly everything seems to be a polyploid. So maybe all of the flowering plants are polyploids. And in fact, the question that we maybe should be asking is, are there any diploids in the sense of a true diploid that hasn't gone through some sort of genome duplication? And then that leads to what maybe the real question should be that we should be addressing, and that is how many rounds of genome duplication or polyploidization have occurred in various lineages of the flowering plants. Now, so far I've been talking about plants, and I told you earlier that there was a good possibility that maybe we shouldn't be thinking about this as just a plant thing. So I'd like to spend just a minute going over what some of the evidence is for the broader systematic importance of polyploidy, or in the modern lingo, genome doubling. There have been proposed at least 2 episodes of polyploidy in the ancestry of vertebrates. In addition, within vertebrates, there are also evidences of polyploidy. One of the best studied examples is in the salmonid fish. And it's also been shown that the genome of yeast, representing the fungi, was also anciently duplicated. So it turns out that these sorts of processes that we understand fairly well in plants, because people have been interested in them for a long time in plants, also apply to a number of other major lineages of eukaryotic life. So what this is suggesting to us is that maybe we need to have a new paradigm for our understanding of polyploidy. And the first point here that I'd like to make is that polyploidization is prevalent, and it's a major force in eukaryotes. It's not just something that plants do, but again, it's something that's important for other groups of species as well. Now, of course, if we really want to understand how this mechanism of species generation or speciation, how this works and what its consequences are, it's not very valuable to look at something that had a genome duplication event 100 million years ago. Instead, what we would like to look at is something that has undergone polyploid speciation very recently, and we can start to tease apart what the contributions of polyploidy itself are versus those of selection over long periods of time. So there are, in fact, a number of polyploid species whose time of origin is quite well known. And in fact, these are species that have all originated within the past 150 years or so. Two of these are species that have originated in Britain, and then two of them arose in the Palouse region of eastern Washington and adjacent Idaho. Now, it's pretty unusual to know the time of origin of any species, so you're probably wondering how on earth have we been able to pinpoint these origin times. And the reason is that all four of these species involve an introduced species. So something was brought to a new location through human activity, and there's a record of when those species were introduced. And because of that, we're able to say, well, the species couldn't have been around before such and such a date because the diploid parents were not present in that location. So these Tragopogon species are going to be the focus of the rest of my presentation this evening. And what I'll be doing is, first of all, giving you some background information on their origins, and then go from there into what we think some of the genetic consequences are of this mode of speciation, and then how that might play into their long-term evolution of species that arise in this manner. So here we have them, our two Tragopogons. This is Tragopogon mirus and Tragopogon macellus. So, as I said, these species originated from diploid parents that were not native to North America. And in fact, the three diploid species were introduced into North America at various times in the 1800s and early 1900s. They were introduced into the Palouse region in the early 1900s, and we know that from herbarium records. So if there hadn't been such a great herbarium there at Washington State, it wouldn't have been known when those species actually arrived there, and it would be a little bit more difficult to pinpoint the time of origin. But this is one of a couple of places where I'll be mentioning the herbarium at WSU and the importance that herbaria and museums can play in this sort of work. So these three diploids, dubious, porphyrius, and pratensis, occur natively in Europe, and they were introduced in the early 1900s. Now, dubious, the first herbarium record for that is from 1928, and so if you figure, well, it may have been around for a few years before any of the avid botanists of the region managed to make a collection of it and put it in the herbarium, we're still thinking that this couldn't it probably didn't get there much more than 10 to 15 to maybe 20 years prior to the 1928 herbarium specimen. So these two species, mirus and macellus, were named and described by Marion Ownby in 1950, and Ownby was the director of the herbarium at what was then Washington State College. He did some exquisite work to describe the ancestry of the tetraploids, and since his early work, there have been a number of studies. Basically, anytime a new technique in chemistry or molecular biology has come along, someone has investigated these species to either test the ancestry further or try to get more information. So there's a wealth of information about them. And again, these species are native to North America. They don't occur in Europe, even though the diploid parents do, because in Europe, the diploids are not sympatric, except in just a few very rare cases. So that's our cast of characters. And here they are, a little more artistically, our Tragopogon triangle. This is dubious, one of the diploid parents, Pratensis and Porifolius. And these two cross to yield Tragopogon miris, which clearly morphologically combines the purple ray flowers of Porifolius and then the yellow center part from Tragopogon dubius. And I should mention, it's probably obvious at this point, but these are members of the sunflower family. They're closely related to lettuce, and in terms of growth habits, they look like giant dandelions. This is Tragopogon macellus over here, and it had these two yellow-flowered species as its parents. Okay, so some people, not too many people, but some people wonder how related or closely related the parental species are, and so I've just inserted this phylogenetic tree to show you what we're finding in terms of the evolutionary history of the diploids. The genus itself consists of about 150 species, and there are these parental diploids are fairly distantly related in this phylogenetic tree. Many of you have perhaps visited the Palouse region or have relatives or friends in that area. It occurs in eastern Washington there, and then adjacent Idaho. And this shows some typical habitat for these species in this area. Generally, they're in disturbed sites along railroad tracks, along roads. Occasionally, they get into native vegetation, but they're mostly in these disturbed sites. Now, Ownby made a number of really quite interesting comments in some of his early papers. He described the populations of these newly formed tetraploids as small and precarious. Some of them, there are only a few populations of each species that he could find at the time that he discovered them, and there were, in some cases, fewer than 100 individuals in a single population. He noted, though, that they'd attained a degree of success, as he called it, and that they appeared to be competing successfully with their diploid parents. And he thought that it would be important to follow the ecological development, as he referred to it, of these newly formed polyploids through time. So 40 years after his naming and describing of these species, a postdoc of ours, Steve Novak, and Doug and I made a trek throughout eastern Washington and adjacent Idaho, this area shown here as on the county level, and we looked for all of the populations of these species that we could find, and we then took a census in terms of the numbers of individuals. So whereas when Ownby first found these species, they were located in Pullman, which is right here, and Moscow here, and over the course of his working on them, a couple of other locations there. But by the time we did this survey, the squares represent macellus and the diamonds represent miris. You can see that there was quite a bit of range expansion that occurred over this 40-year period. And this hatched area here around Spokane basically extends from west of Spokane, essentially at the Spokane airport, along the I-90 corridor, clear as far as Coeur d'Alene. And basically anywhere we found a vacant lot, there were Tragopogon macellus individuals in there. So we estimated that the population size through this huge area here was probably at least 10,000 individuals. So these plants have really established and are essentially outcompeting their diploid parents. One other thing to point out just about the biology of these species is that the tetraploids are shown here as macellus and miris, are much larger than the diploid parental species. And this is something that you typically find with polyploids. They're larger than their diploid parents. Another thing that Ownby proposed was that there were these two different morphologies of the inflorescence, or the head, the flowering part of Tragopogon macellus, and he suggested that these had actually originated through what are referred to as reciprocal formations. So in one case, Dubious here served as the maternal parent, the mom, and Pratensis served as the pollen parent, or the dad, and in that combination, you got what Ownby referred to as the long liguled form. And you can see that these ray flowers here are much longer than they are, for example, in this one. These are taken side by side next to each other. And this particular population occurs only in Pullman. There's just one location where this particular form is present. In contrast, when Pratensis is the mom and Dubious is the dad, you get an inflorescence type where the ligules are much shorter, but where also the head is much more closed. It's kind of cup-shaped rather than open like, say, a dandelion would be. And so you get this very distinct morphology that's due, according to Ownby, just to this reciprocal formation. And this is a morphological difference that you can clearly see at 60 miles an hour as you're driving down the highway. So this is a very good character. In fact, it's so extreme that if you didn't know the history of these things, you might actually wonder whether or not these might be different species. Well, Ownby had no real way of testing that hypothesis of origin except to recreate those crosses. And those crosses only kind of work in one direction unless you're really lucky. So he wasn't actually ever able to carry out the appropriate test of that in detail, but we were able to use a molecular marker, the chloroplast genome, which is maternally inherited, to test that hypothesis. And without going through any of these details over here, this is just a gel to show you that we really did this work. It was clear that this long liguled formation arose as having Dubious as the maternal parent, and the short liguled one had Pratensis as the maternal parent. So we were able to confirm Ownby's hypothesis about that. So in fact, this one species that originated recently formed not once, but twice within that very short time period of probably early 1900s up until the point when Ownby found these species in 1949. So this is what we refer to as multiple origins of a polyploid species. And in fact, there's a lot of evidence from different sources from different types of data for multiple origins of Tragopogon macellus. And when you couple that information with the geographic distribution of macellus around the Palouse, particularly where it occurs with both of its parents, you might hypothesize as many as 21 separate origins of this species, maybe only around 5 or 6 if you just take the genetic information into account, but certainly quite a few separate origins. Likewise, Tragopogon mirus also has arisen a lot of times in this very small geographic area and over this short time period of, at the most, probably 80 years. Now, these species have also been reported outside of the Palouse. Ownby ended up finding populations of macellus near Gardner, Montana, and Sheridan, Wyoming. And then in the 1970s, both of these tetraploids were also reported from Flagstaff, Arizona, along with their diploid parents. We've been able to find the diploid parents of macellus here in Gardner, but there's no macellus there now. But we do have samples from Sheridan, and we're doing some work on both those and the ones from Flagstaff. So in contrast to the way that Darwin viewed speciation, and how I think most people tend to view speciation as there being a single origin of species, these studies of polyploid species suggest that maybe we should be thinking about origins of species rather than a single origin of the species. And in fact, if you take a look at floristic areas on a worldwide scale, you can find the same sort of thing happening. This is particularly prominent in the Arctic, where as you go around, taking a view from above the North Pole, as you go around the Earth, there are many examples of what appear to be the same species having originated multiple times throughout different parts of the distribution. And this is true in just a number of these examples shown here. So multiple origins of these polyploids seem to be the rule. Almost all polyploid species that have been examined are actually of multiple origin. And this can occur at a very high frequency and over a short time period. Now, we've been fortunate enough to have some people in our lab over the last few years who are really a whiz with new molecular techniques. And Von Simons, a couple of years ago, started developing some microsatellite markers in our lab for Tragopogon. And one of the things that I'd just like to point out here is that these are DNA markers that are absolutely excellent for fingerprinting individuals and also populations of plant species. And if you'll just bear with me a little bit, I think this is such a cool example. Oops, wrong one. We'll just look at the left panel over here. This top row is just shows these two patterns that you get in Macellus. And one of them is contributed by Pratensis, one of those diploid parents. And then we were curious as to which population of Dubius might have contributed this other set of peaks. So we looked at a couple of different individuals from different locations, and, well, this one matches that one, but this one doesn't. So we were able to determine that the parents of this individual, this tetraploid, were this individual of Pratensis and this individual of Dubius. So these markers have been really valuable in helping us to identify the parental populations, and as a result, they've actually allowed us to infer additional origins of both Tragopogon Macellus, which is shown here, and also Myrus. And in fact, we've been able to actually make some inferences about seed movement and gene flow among populations using these markers. So this is the output of a computer program called Structure. Oops, I keep doing that. And all you need to notice here are these different bands of color. Each band of color represents a number of individuals from a different location. So this, for example, is the little town of Oaksdale. This is another population from another little town, or from Oaksdale as well. And then you've got a couple of populations from Pullman, and then some of the other little towns from eastern Washington. And the amazing thing here is that generally when we look at these populations, all the individuals in the population have the identical genotype. But when we looked at this population, there's one individual, quite obviously here, that seems out of place. And in fact, what this indicated to us is that it's an individual that originated in this population and must have migrated through a seed dispersal into this other population. So this little bit of gene flow is occurring within Pullman. Lee, you might be able to find where your house used to be. We lived like down over here somewhere. This is downtown Pullman. This arrow right here points at one of the spots where Ownby first found Tragopogon myrus. And these two stars show those two populations, the red population and the blue population. And what seems to have happened is that we've had some seed movement from here over into this population here. So these new sensitive markers are allowing us to make even greater inference about the number of independent origins of these tetraploid species. And these origins, sometimes we've had multiple origins even within a single one of these small towns. So that's some data from Myrus. The same sort of story is true in Macellus, where there are two origins of Macellus within, again, in this case, a very small town, a town that has fewer than 1,000 people in it. And that just summarizes that as well. So what all of these studies combined are telling us is that both of these two tetraploid species have originated multiple times. And so the question then is, well, what does this mean? Does this mean anything for the evolution of the species? Well, for example, if we go with the traditional model of species A crossed with species B, that occurring a single time, we get a genetically uniform polyploid species. And that polyploid species might then have difficulty adapting to any changing environments. This would be the perspective that Stebbins and Wagner thought of as being an evolutionary dead end. In contrast, though, if we have diploid A with a bunch of genotypes, diploid B with a bunch of genotypes, and they form polyploids, and then those polyploids cross with each other, we're not getting a single genotype, we're getting a whole array of genotypes. And by having all of these genotypes, there's a lot more material out there for natural selection to act on. And this may be a way of that ultimately ensures at least a longer-term survival of the species compared with this more traditional model. Now, we're referring to this as the Groundhog Day Hypothesis. So for those of you who have seen this movie, I think you'll know what I mean. For those of you who haven't seen it, I'll just tell you that Bill Murray wakes up basically every day and has to repeat the day till he gets it right. And so there's this whole theme of repetition. And what this seems to us is not that these species are trying to get it right or anything, but evolution is definitely repeating itself. So my second big point here then is, in addition to polyploidization being quite prevalent, polyploid species usually have more than one origin. And not just maybe two origins, but probably, when you look at things with the sensitive enough markers, lots of origins. Now, at this point, I want to turn, don't get scared by this huge list. I'm not going to cover all of these things. But I'm going to turn our attention to some of the genetic consequences of polyploidy and look at this basically from a genome evolution perspective. And we're going to talk about a number of things that are going on within Tragopogon, and where possible, I'll relate these to some other species as well. So the first thing that we've observed in these tetraploid species is something called concerted evolution. And this occurs in what are known as the ribosomal RNA genes. Now, these genes are fundamentally important to life. They have really important roles in the ribosomes. They're present in thousands of copies that are linked up end to end like this. And all of these copies are essentially identical through this process called concerted evolution. Now, in a tetraploid, we would expect the tetraploid or other polyploid to combine the ribosomal genes of the two parents. But when we actually look at this, and there was some great work published in the mid-1990s, particularly from Jonathan Wendell's lab, that indicated that some polyploids actually only have one of these types. They don't have both types, as you would expect. And the inference then is that concerted evolution has occurred. The genes of one species have been converted into the genes of the other. So the question became, though, how quickly does this occur? Is this something that happens rapidly, or does it take a long time to occur? And then also, do you get the same outcome in different polyploids of independent origin? So to just show you what we're talking about, let's look here at one diploid parent. It's got a black peak here. That's a G. We don't have to worry about what the peaks are, but this one has a C. And when you combine them in the tetraploid, well, you get a prominent blue peak there for the C, but underneath that, there's also a little bit of the G. So in Tragopogon miscellus, we actually see both of those. But we're interested in what that might look like on a different level. So this is just a different way of looking at the same thing. And what we could see was that in hybrids, the two contributions, portfolius and dubious, are there in about equal copy numbers. The banding intensity is about equivalent. This is the portfolius band and the dubious band. But when we look in real mirrors, we don't get kind of equal copies like we do in the hybrids that we made, but instead we get mostly portfolius copies and very few dubious copies. And when we do the same thing, we look at the same thing in miscellus, we get the same situation. Mostly pretensus copies and fewer dubious copies. So we thought, let's take a look at this a little bit more quantitatively. And so we looked we counted clones. We cloned them, and we actually looked at the number of copies that we had of one species versus the other. And without belaboring all of this, you'll see that in almost all cases, there are fewer dubious copies than whatever the other parent was. And this is actually some work that a couple of undergraduates did, Annie Sherwood at Washington State and Jenny Roca at Florida. And in all of these cases, dubious, this yellow-flowered species, has its copies lost. So this suggested to us that, well, at least over the span of however long these species have been around, there's been a loss or conversion of these copies. But let's go back to the source, and this is where the herbarium comes in again. And we're going to go back to some 1950 collections and also a 1953 collections, and that's the reason for the car, if you're wondering why there's a car here, 1953 Thunderbird. So the first thing we looked at was some of the Tragopogon miscellus that was collected in 1950 by Ownby himself. And when we looked at these, we found out that the two copy numbers, the two copy types are in equal proportions. Same thing's true in Amiris that was collected in 1950. And the same thing's basically true in this 1953 collection. So it looked like when these polyploids were younger, when they were closer to their time of formation, they actually had equivalent numbers of these two parental types, but that over time there's been rapid concerted evolution. In other words, we've been catching concerted evolution in the act, so to speak, and that this is a rapid process taking potentially less than 60 to 80 years, and because these plants are biennials, that's only 30 to 40 generations. The other thing that's pretty interesting is that this seems to be occurring in the same direction. In almost all cases that we've looked at, it's been dubious that's been the one that was converted into the other species. So again, this looks repetitious, so this is a little Groundhog Day icon again. Now, the really weird thing comes out of some work that some collaborators of ours in the Czech Republic have recently found, and that's that despite the fact that there are fewer copies of the dubious type, it's the dubious type that represents almost all of the ribosomal RNA that gets produced. So, despite having fewer copies, the dubious type is actually the most active in the cell. So there's something very strange that's going on within these new polyploid cells. It's like they're still trying to sort out who's going to dominate, what's going on here. So we've lost dubious copies of genes, and yet the smaller number of genes is actually more active in producing gene products. So these expression studies, as limited as they are showing that in separate populations of both of these tetraploids, the dubious units are highly expressed despite their low copy numbers. And we're just starting to look into this in a little bit more detail to see what might be responsible for these expression changes. So back to our list here. So I've just shown you some evidence for concerted evolution and also a little bit in terms of a shift in gene expression. And I want to just talk a little bit now about some evidence that we found recently for gene loss and basically what we refer to as the downsizing of the genome. Loss of DNA in a tetraploid that leads to a smaller genome than what you might expect if you just added the two diploid genome sizes together. Now, there's been some fantastic work done on tobacco, which is an allotetraploid, in which, through chromosomal study, there's been evidence for lots of genome rearrangements in the tetraploids. And we were interested in whether these same sorts of large-scale genomic rearrangements occurred in tobacco, or occurred in Tragopogon. And so we applied some of these methods. This was work that a postdoc, Chris Pierce, did with us. And this was while we were on sabbatical at the Royal Botanic Gardens at Kew. And we did some work with some colleagues at Queen Mary College in London. And we found, this is just a series of karyotypes of the diploid species, but we found no evidence for this sort of large-scale rearrangement of the genome as it's been observed in tobacco. But we have, over the last few years, found out that, in fact, there have been some genomic changes, even in this small amount of time since these Tragopogon tetraploids were formed. So we used a method that I won't really discuss, except to tell you that this gave us a way to identify a series of genes. And this is a list of those genes here. And we weren't particularly interested in what these genes were. We just wanted to get some genes that we knew were expressed within the tetraploids. And these genes represent a bunch of different cellular processes. Now, we use these genes in what's called a CAPS analysis. And I'm not going to go over details or anything here, but basically this is a way of looking for presence or absence of loss of genes through a very simple cutting of the genes as markers. And what we found was that there were 7 of the 10 genes in which we found a loss of one of the parental genes in the tetraploid, and then another gene in which there was a change in the in gene expression. So again, this just summarizes this. We lost one of the parent genes for 7 of the 10 that we examined, and in one other one there was a shift in expression. So out of 10 genes, 8 of them had some novel property in the tetraploids, again, even though these tetraploids are very young. And this just shows these again in a little bit closer up. And again, we don't need to go through the details, but there's evidence again for gene loss. So, and the interesting thing is that that was true not just within a single population, but the genes were lost, essentially in the same genes were lost in different populations of the tetraploids. So, again, we've got the Groundhog Day scenario. And this shows, again, gene loss in the 3 additional populations of Micellus, so that ultimately there were 5 populations that showed the same pattern. The same thing also occurred in Tragopogon mirus, and this just highlights a couple of those changes. And then in addition, this gene in particular was the same gene in which Tragopogon dubius was the lost gene. And I'll point out here that, remember, dubious was the one that lost those RNA genes. The dubious copies of these genes are generally the ones that are lost as well. So we have a number of projects ongoing in Micellus. We're increasing the number of genes, but we found that the same genes undergo loss in these 5 populations, and the same genes show no evidence of loss. And that loss is usually in the same direction, that is, that dubious is the one that's lost. Oops. So again, Groundhog Day. So of course we're interested in how fast this sort of thing occurs. Is this something that takes 30 generations to start happening, or is it something that happens right away? And so we've looked at F1 hybrids that we've made, and in the F1 hybrids, everything is additive. So no changes right away. It must at least take polyploid formation and then ultimately some amount of time after that. So coming soon are some synthetic tetraploids that we've made. This is work that Jennifer Tate did. She started these last spring or summer. They've been growing. They are now shown here. The plants at a young stage in the greenhouse, they're now flowering. They're several feet high, and this is a picture of the first set of seeds from our very first synthetic tetraploid Tragopogon micellus. And these are some of our fearless greenhouse workers. This is our niece, Carrie, who is a junior at UF. Andrew Doust, who's a postdoc in the lab, and Allie Bigger, and another undergrad who is hoping to go on to med school. Now, all of this kind of repeated patterning is suggesting that there may be some rules to genome evolution, particularly when it comes to polyploidy. And there's a really interesting paper published by Andy Patterson's lab last year that showed that these different groups, this is Arabidopsis, rice, yeast, and a fish, that these have some very similar sorts of genes that they've maintained in duplicate. Incidentally, all of these are ancient tetraploids of one form or another, even though everyone thought they were diploids. There are certain genes that have been retained in duplicate and other ones that have been lost. And we're using the information from this study to start looking at the Tragopogons to see if there are even additional patterns, perhaps rules that control how these genes are maintained. So here, I'd just like to summarize this last bit of the genomic information. These polyploids are evolutionarily dynamic at the genomic level. They're rapid changes, and these include things like genome rearrangements. We have tremendous numbers of gene loss. We were astounded at all of these examples that we found. And also, there's some changes in gene expression, again, in a very short period of time since these species formed. So just a few hints as to what we're going to be working on next. We want to continue with the survey for genes, follow some of these synthetics through multiple generations and see what happens. And then ultimately what we'd really like to be able to do is take all of this information back to the field and do some ecological studies and see if there are some sorts of associations between particular sorts of gene presence or loss and ecological traits. And then one thing that's been particularly exciting is that I mentioned we made these synthetic tetraploids. Well, one of them that we made is what we're jokingly calling Tragopogon floridana. This is a tetraploid that arose between Porifolius and Pratensis, and we made this in our greenhouse. And we're very excited to find out what this does genomically, because this is a tetraploid that has not been reported from nature, and although there are some hybrids between these two species. So we're curious to see what happens when you don't have dubious as one of the parents, the one that's always lost, which one of these two will win out, or will both of them be maintained. And then we also want to take this back to Eurasia, back to take the whole system back and start looking at some of the polyploids that are in Europe. The ancestries are mostly unknown, so we're working on that at the moment. But what we are finding is that a few of these species, we know what their parents are now, and that forms a really nice continuum in terms of ages. We have our triangle here with our new floridana inserted. We can look at F1 hybrids. We can look at our resynthesized tetraploids, the tetraploids that occur in North America, and then much older Eurasian polyploids, and see what sorts of things are happening, if we see the same sorts of evidence for gene loss or maybe more extensive gene loss in the polyploids that are much older. So I'd just like to conclude with a few major points, and that is, first of all, that polyploidy, as I mentioned throughout, is a major force in plant and now animal and fungal evolution. Speciation through this mechanism is not what Darwin had in mind. Instead, speciation is instantaneous, and it occurs through the bringing together of different genomes. And the question, of course, that we've been wondering is, are there any diploids out there? Tragopogon we're really happy with as a model for studying this recent polyploidization phenomenon. The species originate multiple times, frequently on a local scale and over a short time period. There are some potentially very important genetic consequences. We've got concerted evolution in the act, and we've got evidence for gene loss. In both of the tetraploid species, the same genes show loss across populations. It's usually, again, the dubious copy that gets lost. And very importantly here, in terms of both the gene loss and the origination, evolution seems to be repeating itself, giving us the opportunity to have replicated experiments that nature's made for us. So I'd just like to acknowledge all of the collaborators in this work. First of all of this work's been done in collaboration with my husband, Doug. And then this is a majority of the Tragopogon team up until last fall. Jennifer Tate was a postdoc with us for 4 years. Vaughn Simons for 2 years. Both of them have faculty positions at Massey University in New Zealand now. Jean Coe is a graduate student working on Tragopogon miris, and David Lefkowitz was an undergrad who's now in med school at UF, but we never see him even though he's just down the hill because he's always busy studying. And then these are some of our European collaborators. They sent us this picture, and we thought, this is just great, so we use this one all the time. Andrew Leach is our collaborator at Queen Mary College in London. Jung Lim is the one with the magic thumb who makes the beautiful chromosome squashes and does the chromosome painting work. And then Alish Kovarik is the head of a big lab at the in Brno, Czech Republic, and Roman Matyasik is one of his graduate students. And I think that's it. So thank you very much for your attention. I hope people weren't sound asleep the whole time after having that wonderful large dinner before I started talking. So anyway, thanks very much for your attention. I'll be happy to answer any questions if people have them.